Hello everyone, welcome to the first episode of my video series on medical and biological datasets for artificial intelligence. In this series of videos, I will discuss about the methodology and the mechanism of creation of these datasets and the challenge, opportunities, and also related works and the application of these datasets for AI scenarios, artificial intelligence. I am Hamidreza Bolhasani, Chief Data Scientist and founder of DataBiox, which you can see here, and we will be more familiar with that in the next slides. Okay, so first of all, let's have some definition about AI, artificial intelligence. Maybe almost all of you know what AI is, and these days is very trendy, but let's have a very brief review. And the slides are taken from the Stanford University, as you can see here, the reference. Okay, AI is not a new thing. It refers to more than, as you can see here, 7 years ago. Okay, the first definition of AI, artificial intelligence, is based on John McCarthy definition, the science and engineering of creating intelligent machines. Okay, one of the leading subtypes of AI named ML, or machine learning. Arthur Samuel in 1959 has a very nice definition for ML. ML is a field of study that gives the computer to learn without being explicitly programmed. Okay, in the traditional programming, you need to have data and some rules to get answer. It is traditional, but based on AI, you have some data and also some answers or label, and you will extract some rule. This is the new era, okay, named machine learning. And subtype of machine learning algorithms are brain inspired algorithms. Brain, it's not the meaning of just the human brain, no, biological brain. An algorithm that take its base functionality from our understanding of how the brain operates. We can conclude that AI is a combination of engineering and also neuroscience. Neuroscience will help us to get better understanding of how the brain works, and engineering and mathematics will help us to model these understanding, and it works very well until now. Okay, brain-inspired algorithms are categorized in two categories: spiking neural networks, SNN, and artificial neural networks. We will not discuss about spiking neural networks right now, and the main focus will be on artificial neural networks, and especially deep neural networks. So what is the artificial neural networks? Artificial neural networks take its main idea from the biological neuron, as I told to you. The biological neuron of a mice. It's gotten from this book. The reference is here, Deep Learning for Computer Architects, in 2017. As you can see, the main part of a neuron, biological neuron, is axon, cell body or soma, and dendrite. In each decision, in each action, a spike will happen in our brain. Okay, mathematical modeling of this will result in this. This is a artificial neural network with just one neuron. Okay, we will discuss more about this, the function and the mechanism of these networks in some other videos, but right now our focus is on just introduction and going through the introducing the medical data sets. Okay, just have these definitions in your mind and keep in your mind that artificial neural networks is an artificial network of neurons that is taken its main idea from the biological neurons. And what are deep neural networks? When the number of layers became more than one hidden layer, it named deep neural networks. You can see many deep neural networks right now have several thousands of layers to extract more features from the inputs. Okay, and one of the most successful and famous neural networks that these days really help for specifically for image problem, also in medical and biological, are convolutional neural networks. Also we will introduce them in some other videos, but right now please keep in mind that convolutional neural networks, that very simply their mechanism is based on getting the similar patterns based on some mathematical functions, are very popular for image recognition. And in medical and biological data sets, like as you can see here, a histopathological sample, one of the main problem are image classification or signal detection. Okay, but image are very hot, and also videos these days for some problems like endoscopy and colonoscopy, which also will be covered in the next couple of videos. Okay, so what does a deep neural networks needs to be trained? We need data. Why I write this big in gray? Because we don't know exactly how much data we need. We need these data as much as the network trained well, and we got and we reached to the accuracy that is suitable for us. So there are many types of medical and biological images that you see some samples here. In this video series I will introduce one of these data sets, image data sets, in each episode. Okay, for example, histopathological images, CT scan, MRI, PET scan, positron emission tomography scan, endoscopy, colonoscopy, some genome information, and many other things that will be covered later. But the first data set that we will cover here, and one more thing, data sets are becoming very important and popular these days because everyone is talking about the AI, artificial intelligence, deep learning everywhere. Okay, but it is very nice, but how we can feed an AI network with data? So data sets are really important. There are lots of databases that you can refer to them, like Google Datasets, Kaggle, many prestigious institutes like National Institute of Health, National Cancer Institute, also Mendeley from Elsevier publication. Mendeley database is really very good, and we our team have published and uploaded many data sets on that. Harvard Dataverse, which you know that's really prestigious, Carnegie Mellon, University of California, Irvine, and many other universities are also DataBiox that are founded for AI and data-driven solutions for healthcare and medical sciences. And as you can see the slogan, we give life to the data, and this is my mission. Okay, but let's cut the introduction and go through the first real data set. In 2020, with this team, you can see, and the help of The main idea was from this prestigious pathologist, Dr. Elham Amjadi, and this surgeon, Dr. Mariam Tabatabayan, and she's also my PhD advisor, was histopathological image dataset for grading breast invasive ductal carcinoma. This paper thankfully got really notice on that. I've received many requests from all over the world, from prestigious universities and institutes, for accessing these datasets, and also we got some rewards on that from Elsevier and other institutes. For example, one of this article were linked to the United Nations Sustainable Development Goals, helping to tackle some of the world's greatest challenge. Okay, but how we produce this dataset, and I also will put the link of this paper. If you want to read and download it, I will put for you. Okay, and before introducing the dataset, let me show you something. This is the data. This is the DataBiox site. Always I introduce the new datasets in this site. Okay, and also you can see our projects here, and if you are interested in any datasets, you can go to the datasets menu and select your required dataset and fill this mandatory information and submit this one thing. And this is the paper that is published in Elsevier, and we'll put the link for you. Okay, let's go to introduce this dataset. At the first step is selecting the patients diagnosed for this specific problem, diagnosed with invasive ductal carcinoma. So at first, for any data sets that we want to create, at first we should select the scope. So here the patients diagnosed with this. Collecting H&E stained slides. You know that histopathological samples are strained by hematoxylin and auxin, H&E colored in a glass. Okay, and then labeling these by a pathology specialist. In this case, there are 3 grades, as you can see, so 3 label. This is a supervised machine learning problem. Supervised learning, sorry, not machine learning, because it's the problem will be deep learning because it has many features and it's based on the image. Okay, it is supervised learning. We have data, and also we have the labels. So we have in this problem we have 3 grades, as the pathology experts know that based on the standards, based on the books and references, invasive ductal carcinoma are diagnosed in 3 grades, and grading is very important for also treatment plan and also prognosis and many things. Then microscopic imaging is done. In this project, we got sample in 4 magnifying power: power 4, power 10, power 20, and power 14. Okay, 40. Sorry. I will show you then that some newest articles show us that this in breast cancer, invasive ductal carcinoma, this power 40 for classification have the highest accuracy. Based on a paper in IEEE Access, it's about 100%. But I will also share the link of that paper with you. Okay, so, and then organizing the dataset, publishing and organizing the dataset. I will show you the dataset, putting them into different folders, and also with the labels, and also making sure that no information from the patient is there. It is private and the privacy is kept. Okay, in this work we can see that for the number of patients in grade 1 we had 37 and 259 images. For grade 2 we had 43 patients and 366 images, and for grade 3 44 patients and 297 images. And also in each grade you can see the number of photos in each magnification level. That you can see 40x has the highest number of images, because as I told to you, it has more accuracy based on some research. Okay, so also let me show you the paper. Okay, here you can see that this is the paper, and at last we can see the number. Yes, we can see that totally we have 124 numbers of specimen. We have totally 124 samples, which in each power we have these samples. Okay, and totally we have about 1000 samples in this data set. I think 922. Okay, and also let me show you that paper that shows us the 40x is the highest accuracy. As you can see, based on this paper published in Frontiers in Genetics, deep learning based analysis of histopathological image of breast cancer in 2019 published, we can see that 40x is shown very good and high accuracy. Okay, and also we got to this result. Also we got this. Okay, so also about the data set, you can see if you download it from if you want to access this data set, you will fill this form, and I will send you a link that is linked to Elsevier Mendeley Dataverse. And when you download it, you have 3 folders. The first folder is related to invasive ductal carcinoma grade 1, so this is the label grade 1. You can see we also have 4 level of magnification, 4, 10, 20 and 40. We can see that all of the images are seen here. Okay, let's show you, because it's based on cloud and maybe slow. Okay, it is 4, it is 10 x magnification level. Sorry, it's based on the cloud, and maybe, oh yes, a little slow. This is very nice. 10 time magnification, and also 20, and at last 40. Okay, we can see this. Okay, it is a good data set for also both of the groups, both for pathology specialists for educational purposes, because these are labeled by some very expert pathologists, also for engineers, artificial intelligence experts, to develop and produce new algorithms to get high accuracy and helping the medical society for doing better decisions based on this. And also this is grade 2 in the same way, also grade 3. Okay, it was the first data set that I wanna introduce to you. I hope this would be informative and useful for you, and if you liked it, please don't forget to subscribe in my channel and kindly introduce it to your friends and colleagues, and also stay tuned for the next videos on introducing new medical data sets that's provided by us or other institutes, and also the new training videos for classification, for doing some great jobs and solving problems. Okay, hope to see you next videos. Bye.